Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome to The Explainer. Today, we're diving into one of the most exciting, most revolutionary frontiers in all of medicine, the ability to rewrite our own genetic code to cure disease. I mean, think about that for a second. What if we could cure diseases not just by managing symptoms, but by going right to the source and rewriting our very biological code? Well, this isn't science fiction anymore. It's the incredible promise of cellular and genetic therapy, a totally new approach that tackles illness at its absolute root. So to really get how this is even possible, we need to break down the fundamental tools of this new medical era. It really all starts with two core concepts: cell therapy and gene therapy. And it's a really crucial distinction. Okay, so think of cell therapy as using a whole living cell as the treatment. It's like a tiny biological worker sent in to do a job. Gene therapy, on the other hand, is more like a molecular surgeon. It uses DNA or RNA to make a super precise fix inside our genetic instruction manual. But, where this field gets truly powerful is when these two ideas merge into what we call gene-modified cell therapies. This is where we take a patient's own cells out of their body, give them a kind of genetic upgrade in the lab, and then put them back in. The best example of this is CAR T therapy, which basically trains your own immune cells to become, cancer-seeking missiles. It's amazing. Now, this whole kind of complex field, it really just boils down to two main strategies. And the difference between them comes down to one simple question: where does the genetic fix actually happen? Does it happen inside the body or outside in a lab? And this right here perfectly illustrates those two fundamental paths. On the left, you've got in vivo therapy, which means within the living. That's where the genetic medicine is delivered directly into the patient. On the right, we have ex vivo, which means outside the living, where we take cells out, modify them in a lab, and then put them back. The choice between these two really depends on the disease and what part of the body we're trying to fix. All right, so let's explore that first path, the direct approach, what we call in vivo therapy. This is where the treatment is delivered straight into the body to work its magic right there on site. The best way to think about this is like a super sophisticated biological delivery service. A microscopic vehicle, which we call a vector, is infused into the body. It then has to navigate all the way to the target organ, find the exact cells that need help, and then, boom, deliver its therapeutic genetic payload. It's a mission of just incredible precision. So this direct delivery method is perfect for organs that we can't just, take out and put back, like the brain or the liver. The main tool for the job is something called an adeno-associated virus, or AAV. Now, it's a re-engineered, totally harmless virus that just acts as the delivery truck. And crucially, it delivers the new gene as a separate piece of code called an episome. You can think of it like a temporary software patch rather than a permanent hardware change, which is great because it lowers the long-term risks. But there's a big catch. A lot of us have already been exposed to similar viruses, so our immune system can spot this delivery truck coming and destroy it before it even gets to its destination. And listen, this isn't just theory, this is happening right now. These are real, life-altering therapies that are already approved. Luxturna is literally restoring sight to people with a form of inherited blindness. It works by delivering a correct gene directly to the eye. And Zolgensma treats a really devastating neuromuscular disease with a single IV infusion. I mean, this is medicine that was once the stuff of pure science fiction. Okay, so now let's turn to the second path, the ex vivo approach. Now, this method is a lot more complex logistically, but it opens the door to some absolutely incredible feats of cellular engineering that happen outside the body. This whole process is kind of like sending your cells to a high-tech repair shop. First, they're collected from you. Then they get shipped off to a specialized lab where they're genetically re-engineered and multiplied into an army. While that's happening, the patient often gets chemotherapy to clear out some space. And then finally, the upgraded cells are infused back into the body. It's this multi-step, highly personal process that transforms a patient's own cells into a powerful living medicine. And this repair shop approach has already given us two totally revolutionary types of therapy. On one side, you have CAR T therapy, which, as we said, weaponizes a patient's own T cells to hunt down and destroy cancer, creating what people call a living drug. And on the other side, you've got HSC therapy, which can permanently fix genetic blood disorders by basically creating a lifelong factory of healthy blood cells inside your body. And to make this really concrete, just look at Casgevy, a new therapy for sickle cell disease. It uses the famous CRISPR technology, but not to add a gene. It uses it to knock out a single gene called BCL11A. See, this gene's job is to switch off the production of healthy fetal hemoglobin right after we're born. By knocking it out, the therapy just flips that switch back on. It is such an elegant solution. Instead of adding something new, it just removes a single instruction to unlock the body's own power to heal itself. And the clinical landscape for these ex vivo therapies is just exploding. We've got CAR T therapies like Kymriah for aggressive cancers, which work by adding a new gene. Then you've got cures for inherited blood diseases like Zynteglo for beta thalassemia. That also uses gene addition. And of course, the groundbreaking Casgevy for sickle cell, which uses that gene knockout approach. This is all fundamentally rewriting the future for patients with these awful conditions. But with this incredible power to rewrite our biology comes immense responsibility. These are not simple drugs. They are powerful, often permanent changes. So it is absolutely crucial to understand the risks and the rigorous oversight this whole revolutionary field demands. The main safety concerns really fall into two big buckets. First, there's the risk that your body's own immune system might just reject the therapy, attacking the delivery vehicle or the newly engineered cells. Second, and maybe more serious, are the risks that the genetic modification itself could go wrong. Like the new gene could insert itself into the wrong place and accidentally trigger cancer, or the gene editing tool could snip the wrong piece of DNA. And to guard against these long-term risks, regulatory bodies like the FDA are extremely cautious. For any therapy that permanently alters a patient's DNA, they require patient follow-up for up to 15 years. 15 years! That's how seriously the long-term safety of these treatments is taken. But maybe the single biggest challenge right now is just scale. The current model is what you'd call a batch of one. It's a highly personalized, logistically complicated, and unbelievably expensive procedure for just one patient. For this revolution to really reach everyone, that has to change. So the entire field is racing towards a future built on a few key pillars. First, developing better delivery tools that can act like stealth bombers, evading the immune system. Second, creating safer, more precise gene editing technologies like base and prime editing that can make changes without actually cutting the DNA. And third, which is really the holy grail, is creating off-the-shelf therapies from healthy donors that can be mass-produced, making these cures accessible to everyone. So what's the ultimate takeaway here? We are, without a doubt, in a new era of medicine. These aren't just treatments. In many cases, they are actual cures. The great quest for the next decade is going to be solving the huge challenges of safety, scale, and cost to make these incredible genetic medicines available to every single person who needs them. We've learned to read the code of life. Now we're finally learning how to write it.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.